This is the so-called geroscience hypothesis, that by modulating the rate of aging, you can actually modulate disease and a lot of diseases simultaneously. So is there any reason to believe that that hypothesis is plausible? And it turns out actually there's quite a lot of reasons to believe it. So lifespan, and therefore, based on what I just showed you, the incidence of disease appear to be genetically regulated, evolved traits that are therefore manipulable. And there's multiple lines of evidence on this. So the first is theory. Energy and resources are not infinite. Therefore, every organism has a choice of how it deploys that energy or whatever limiting resource it is, right? Between growth and reproduction and maintenance and repair. And that optimum, which selection will select for, is set by what's going to maximize fitness, not by what's going to maximize your personal lifespan. And the things that maximize fitness are number of offspring, time to them, and the quality thereof. And this theory makes the prediction that organisms in different niches will therefore end up with a different set point, a different optimal lifespan that has been selected for. And that's exactly what we see in comparative biology. Lots of examples of this. I just picked a couple. So the first colorful diagram, which I know you can't read, but that's the radiation of Pacific rockfish. So this is a very new clade of organism. The common ancestor was only 8 million years ago. There is enormous variance in the maximum lifespan across species within rockfish. So some species live 10 years, others live 200 years. Moreover, those differences exactly match what we would predict from evolutionary theory. Changes in depth and size, which correlate with risk of predation and risk of starvation, exactly predict the differences in lifespan. You not only see this across species, but you can see it within species. So the black and white diagram is eusocial insects, so insects that have a queen. In the common black ant, as an example, a worker ant lives 10 months. A queen can live 30 years. They have the same genes, just which genes are turned on and off. So there's a lot of evidence that lifespan is highly manipulable. And then, of course, lastly, a lot of organisms without a soma don't age at all. There's no change in their mortality or reproductive output with age. That's true for a lot of plants. Most organisms that are unicellular that don't have a soma. It's even true of some animals that can revert to a germline state, like hydra and immortal jellyfish. There's a third line of evidence around plasticity based on conditions. So pretty much any kind of insult you give an organism, as long as it's mild, makes it live longer, which seems incredibly counterintuitive until you understand it in light of the theory at the beginning, which is if you're in a famine, it does not make sense to reproduce. So you shift those resources to maintenance and repair, stretch, live a little longer, wait for the next breeding opportunity, and hopefully that next season is a good season. And that's the exact surprising result at the top that was discovered in the 30s that kind of kicked this field off by Clive McKay and colleagues at Cornell. They were working on vitamins and nutrition and got this surprising result that when they fed the animals less, in this case rats, they lived longer. And that's been highly replicated across many labs and many organisms. It's not just lifespan, it's healthspan. So the diagram on the bottom, those are two rhesus monkeys. They are the same age. The one on the bottom has been mildly caloric restricted. You can see it looks much healthier, and it is by pretty much any biomarker much healthier. We see this in humans as well. So the top data are epidemiological data from Phelan and Rose across different populations. A negative correlation between caloric intake and lifespan. That's lifespan data. On the bottom is healthspan. This guy is a member of what's called the cronies, the people who practice caloric restriction with optimal nutrition. And you can see he looks healthier in the second picture, and indeed by pretty much any biomarker is healthier. We see a line of evidence with genetic interventions. So Cynthia Kenyon's very famous paper that a single gene changed could make a worm, in this case, live twice as long. And on the bottom is human data. Actually, human variations in genes that are involved in the same pathway as these worms are associated with centenarians, people that live 100 years or more. And lastly, we have evidence from pharmacological interventions. So we know it's had proven that you can give a drug or various other interventions to, in this case, a mouse, and make it live longer. This is the first proof. This is from the National Institute of Aging's ITP, the Interventions Testing Program, showing that rapamycin extends lifespan. Lots of other examples since then. The only thing on this page that's missing is have we run a long-term trial in humans to see an intervention extend their lifespan? Now, it's hard to do that, right? It takes a long time, it costs a lot of money, but based on the evidence you see on this page, I think you conclude with a reasonable degree of certainty that if were we to do that, we'd also see the human data as well.